Colleagues, ladies and gentlemen, welcome to this pair seminar. My name is Qingyan Chen. I am the director of CUI Academy for Interdisciplinary Research. And today we are very pleased to have Professor Zhanfeng Cui from the University of Oxford to give this seminar. Professor Cui is a staunch friend of CUI Academy for Interdisciplinary Research, or PAIR. He has been the PAIR Senior Fellow and also a member of a PAIR International Advisory Committee. At Oxford, Professor Cui holds the title of Donors Pollock Professor of Chemical Engineering, and his research interest span the regenerative medical engineering and technologies, biomanufacturing, bioprocessing, and bioformulation, and medical devices for detecting infectious disease, including COVID-19 virus. So his research interest is so wide, really span a number of disciplines, and therefore is a perfect example for PAIR to learn. So he is also a very experienced inventor and the multi-patent holder. So Professor Cui developed a range of technologies, including 3D cell culture, bioreactor technology, and the rapid test of COVID-19. Probably many people are still remember when we went through the airport for the fast detection of COVID-19. So Professor Cui's technology was used in the UK airports and also was trialed here in Hong Kong International Airport. So he has developed a lot of enabling technologies for regenerative medicine, and those are really being used for the treatment of cancers, diabetes, neurodegeneration, and musculosis skeletal conditions. So those are just a few to mention that. It's very long, so I'm not going to read all of them. And in addition to that, and his research innovation is, in addition to those innovations, Professor Cui also promoted the engineering and scientific collaboration between the UK and China, for which, and then he was bestowed as the lifetime honor as the foreign members of the Chinese Academy of Engineering, and he's also a fellow of the Royal Academy of Engineering in the UK. And he leads the Oxford Suzhou Center for Advanced Research, or OSCA. This is the only, actually the first overseas centers established by Oxford University. So it's very prestigious. So ladies and gentlemen, without further ado, please join me in welcoming our seminar speaker, Professor Cui.
Good morning, ladies and gentlemen. And it's a great pleasure, actually, to come back to PolyU and to give this PAIR seminar. I think, you see, I really believe in interdisciplinary research, and also my research is all actually in interdisciplinary area anyway. So I like the setup of PAIR, and I think it's actually set a very good model for all the other universities to look into how we cope and how we respond to the changing world. Basically, we live in the modern world, and all the real problem, you can't solve it individually or by single discipline. So that's why interdisciplinary research really is the future. I think that's keep that in mind in our research and also in education as well. So I give this talk, as you see, AI. It seems everybody talking about the AI now, so I must put the AI there. You see, I'm not an AI expert, and so, but we talk about AI, touch a bit, and the biomanufacture. So that is probably to many people not quite familiar, and probably the ATMP is less familiar. most is advanced therapy medicine products. So I think maybe the three aspects I will see through this some examples to demonstrate actually you really need interdisciplinary research can solve the important problems. So I think this Professor Chen gave a nice introduction. Thank you. And people ask me, what do you do? A lot of people come to Oxford, they come to my lab, said, you are the chair of chemical engineering, but what you're doing, nothing to do with chemical engineering. I said, no, that's not right. Because what we do is all chemical engineering, but application of chemical engineering in different areas. So this is actually gave a snapshot of what we do. So I do like chemical processing, membrane separation, energy storage. I have a team working on energy storage, hydrogen production, and but the majority is about the bioprocessing and the medical technology, in particularly tissue engineering and the stem cell technology. So you see, I work in a biopharmaceutical as well, but I'm largely dealing with cells. So then when you do tissue engineering and the stem cells technology, part of that is to use clinically, and the part of that use in vitro models. This is now very fashionable called organoids. And then we used to call it microorgans. This is to develop human microorgan for drug testing, okay, for drug development. So that's basically what I do. But I always say I am a chemical engineer, but working on the disciplinary area. So now we're back to what is biomanufacture? So this is manufacturing. This is what I'm the topic. So it's a use of living organisms. So it has to be living, okay, cells, bacteria, yeast, whatever, and their biological compound to produce commercially viable products. We are engineers, right? Engineers to make it happen. Scientists is try to discover what happened, but we will make it happen. But if you want to make it happen, is engineering discipline, you need to make money. Bottom line, the cheaper products make expensive products through the process, make financially viable and meet the society's need. So it has to be in large scale. So it's not in a test tube. I proved it, but then you have to produce it in large quantity. So this two part, one is a bio part, is a living factory. So it's use living organisms, and then you make a lot. So that's, and make a lot is an industrial process. It has to be in a controlled way. So it's designed to do that, controlled way, make what you want. So that is a manufacturer's part. So combine these two, this is a biomanufacturing. So this is in contrast to chemical industry, you use chemical process, you use chemical reactions, you can make products. Now is use biological cell. So what kind of products are made through biomanufacture? Very common, okay? Wine, okay? Drinking for yogurt, vinegar. So they all through biological fermentation, right? And then you have now a lot of people working on biopolymers. Say use CO2 plus water, use sunlight, you use the bacteria, and they make a polymer. So you don't rely on fossil fuel. So it's all this kind of biological, bio-based materials and enzymes as well, right? You see a detergent, they said we have enzyme in it. So that will clean better. But what I'm particularly interested in is this therapeutic medicine, this area. So this kind of monoclonal antibodies, this is, we all know that, and antibiotics. There's a lot of antibiotics made by fermentation. But there's a new type of thing, new type of AIDS we call the next stage, is the cellular products and the tissues and organ. So can we make human parts, okay, in industry? So this is a new category of medical products. Is basically based on tissue engineering. This is like a growing human tissue outside the body and put it back, put in the implant in the body. That's tissue engineering. And our stem cell therapy. So for example, if you treat a neurodegeneration and our stroke, the probably only way is to use, if you can't use tissue engineering, you have to use stem cells. Put the stem cells into the bodies, and the stem cells will fill the gap, will make connections. So that's a stem cell therapy. And then also gene therapy through editing of a gene to repair some defect. So this new category of products, they are not drug, they're not produced by tons, right? They're not a conventional drug, and they are not devices, because this put in the body, integrate with the body. So it's a new category of therapeutics. So FDA and the EMA, they said, if you try to apply for approval, how we deal with it? So they come with the new category called Advanced Therapy Medicinal Products, ATMP. So this is what I'm trying to address today, how to make this. So tissue engineering is actually, the principle is very simple. It's not that difficult. It's not that. So if this individual patient need a piece of tissue, let's say a piece of bone or a muscle or our soft tissue, due to tumor removal or accident, traffic accident, you need to got a bone fracture. So what you could do is that you take an image, said, I need this particular shape, this piece of tissue. Then you use prototype processing, like for example, 3D printing or any other prototype, make a scaffold like that shape, and you put cells in it, and then keep cells happy, give some growth factors. So put this in the environment, so-called bioreactors. So let them grow. And they will grow, in theory, into the tissue you want. And then you can implant into the body and fill the gap, integrate with body. So basically, we are producing human body parts outside the body. That's tissue engineering, right? So then it looks simple, but then you ask a question, how long I culture that inside? Do I have to get real like a tissue, all the mechanical properties, functional properties, everything like a tissue put in the body? Or I can half down, right? I said, let the body take over. The body, after all, body is the best bioreactor because your tissue needs to be there. So biomaterials people will say, why you need the cells? Because in the body, you have a lot of cells. That's what we have. So you just put a scaffold, put it in biomaterials, no cells, it can provide the support. The human cells will penetrate and integrate. And the cell therapy people will say, why you need a scaffold? Because scaffold, what you use is biodegradable, later they disappear, but the cells will make extracellular matrix, form a scaffold anyway, so we just put the cells in it. And then if you do the culture this way, you can say, I just mix the cells, put it in, or fold it down, put it in. So it's a lot of variations here. So but this idea is proven. You actually can buy skin products at the moment, 5 kinds of products in anytime you buy it, $500 a piece to treat burn and a chronic wound. You can buy it, so it's proven. But other tissues products is a lot of study, a lot of research, but not medically approved. There's various reasons. For example, the most needed products is a small diameter blood vessel. Okay, this is for heart bypass, for people doing heart bypass operation. So if you have produced these blood vessels off the shelf, if the patient come in, open up, sutured, and then that's it. But at the moment, they have to take a piece of vein blood vessels from the patient and the stitch replace that bypass. But FDA still not approved this tissue engineering blood vessel. The reason is that FDA will ask a simple question. How can you guarantee this piece of blood vessel will last for 30 years, 40 years, never break? Because it's so critical. If that blood vessel bursts, the patient will die instantly. So that is the reason they haven't approved that yet. But it's very successful in plastic surgery, right? You know, the people want to I want a bigger nose. I want to get up. So it's actually used widely, and this is commercially very successful. And then for cell therapy, so this is actually hope for the hopeless, right? So, you know, paraplegic patient, they live on wheelchair, quite often due to traffic accident. This is for spine injury. If the spine gets injured on the back, they actually they have to stay in the wheelchair for life. If they actually on the neck, and they have to lie down for life, right? That's even worse, tetraplegia. But now you could actually use a stem cell therapy to enable the paraplegic patient to stand up. And that's a Polish gentleman, and UCL's work. In China, we had wider report as well, and get the patient to stand up, paraplegic patient. But the trouble at the moment is that we have people very responsive, actually stand up, cured, but you have people not cured. You see no effect. So why? So that is actually a lot of fundamentals, a lot of research needed. So the other one's CAR T. So that's a little girl, and she is the first one. I think I got a better picture for that. Okay, so this is actually this little girl called Emily, and when she was 6, she got acute leukemia, basically blood cancer. So there's no cure. Almost say, okay, poor little girl will die. But then this CAR T therapy is basically the first test. She said, well, we will accept this trial. So is get this first CAR T patient treatment, and they're completely cured. Right? This is the start of the CAR T. So CAR T is interesting because actually it's very simple to understand. Actually, most of our people, most of us, we always have cancer cells in our body. So cancer cells is just the cells when they're programming something wrong, get a mutation. You could call it cancer cells because it's not normal anymore. But our body immune system, it's largely relies so-called T cells, and they will identify these bad guys and kill them, clear them, right? But this cancer, blood cancer, these cancer cells, they are very, very clever. When they become out of control, they actually enable themselves to actually camouflage, and the T cells could not identify with them anymore. So they out of control, they grow a lot. Basically, the police or the armed police get corrupted. They couldn't find them, they can't see them anymore. The CAR T therapy is take the patient blood T cells come out, said, okay, you cannot see the cancer cells anymore. Let's train you to see them. So they actually put a so-called receptor to the T cells, and then so they would be able to find the cancer cells. And then in the body, outside the body, we grow a lot of them. Okay, so we have a large army who can identify the cancer cells in, and so we put in the back of the body. So they go to the get the back of the body, identify the cancer cells, kill them off, clear. So that's the principle for that. Okay, so that is proved that, and it proved work, but then it's just very expensive, right? The American ones usually 300. I think a cheaper ones over $300,000 US. And the UK government get a price and 228,000 pounds. And the UK government just say, NHS said, no more than 200 patients per year. Because if you got too many, it will bankrupt the system, right? So it's very expensive. In China, I know this is probably in trial is 1.2 million Chinese yuan for. And so this is come to this. I think this is a stem cell. We can go back. Okay. Now the other example. This is about 3 weeks ago. UCL they reported BBC. Basically, they use gene therapy. Remember, I mentioned that the ATMP is a tissue engineering, cell therapy, and the gene therapy. So this is the first gene therapy treat Huntington's disease. This is the brain, in the brain cells, this is Huntington disease. Basically, inside the brain cells, they have protein deposition. It's not Alzheimer, but Huntington. So they do one treatment, put it into the gene, edited gene, put it into the brain, and they can clear the protein. So this slow down the development of Huntington disease. So that's about 3 weeks ago. But this is all we said, we can do it. But why it's so expensive? If it's too expensive, we cannot treat the most ordinary people, right? We do not want to do something really great, but only for the super rich. So this is actually, so how we get this, why is it so expensive? If you're looking at this, this is autologous cell therapy. Basically take our own body cells, go out to do processing and put our back to our body. This is autologous. And because for allogenic, basically one cell fit all is still not reachable. At the moment, we cannot do that because the body will reject the cells. But it's your own cells is fine. So you take the cells, go through all the different processes, okay? Isolate, purify the cells, then grow a lot of them, cell expansion, and harvest, and then formulate, package, detection, release, and it's a very complicated process. This is, give you a couple of examples. So this is the first generation of chondrocyte therapy, ACI. You know, young people, you play basketball, football, then you don't feel it. When you get old age, 50s, over 50s, your knee will get knee pain quite often because the cartilage damaged. The cartilage sponge actually in the knee, and they actually absorb all the shocks, all the mechanical loading. But then in the cartilage, the cells density is very low, and they do not grow. When you adults, you get a certain number of cells, then they're all there. They just maintain your cartilage. But then if that's damaged, you do not have cells to maintain your cartilage, and it's damaged. So later years, you get pain. So this is the first generation of ACI. Basically, you take patient cartilage cells from no mechanical loading site, take a biopsy come out, and grow a lot of cells and put back to repair the damaged site. So this is the first generation, and then this now improved the third generation already. But you just see this is take the biopsy, send it to the processing plant, take about 3 to 5 weeks, and then you got the cells come back. So what is the patient from the hospital is just a piece of this little tube here, biopsy. What a medical doctor get is a syringe. You can inject the cells. But all this processing here in the middle is grow cells. And all human, you need the people, change the medium, and then you got machine, you need a GMP clean room facility. That's why it's so expensive, right? And then it's very easily get it wrong because you got a lot of human intervention step. Every step wrong is get wrong. So that's why what we want to do, we want to replace this all by machine. If you got a machine, automated system, that would be a big step, can reduce the cost. I mentioned the CAR-T process. The little girl. This is actually what happened. You got this, take a 50 ml or 100 ml blood, and then use all this processing step, and until later put it into the human body. And the step, so many steps, every step you got human involvement, and then every step is a sequential. You got one step wrong, you have to start from beginning, right? So this is every step is very critical. So that's why it's so expensive. So we look into this, how to reduce the cost, where you set up your centers. But finally we said, actually, if you get an automatic machine, and this is later I will say why we need the AI and reduce the cost. That is really the reason engineering contribution. This is to grow human. This is the first tissue engineered products. How to grow human skin, right? Now it's process is complicated, the cell, different type. But what I would like to draw your attention is the timing. You're looking at grow a piece of skin, take 20 days. Take 20 days. Think about the manufacture process we are familiar. Okay, it's just like if you do chemical process, you want the resident time second or minutes, never be days, right? You see, the cell culture that is also takes weeks. This is day. So it's actually the manufacture process is very different from what we are familiar with. So we can do it. We want to do it cheaper. So this is where I think AI can come in. This is why biomanufacturing need AI. So if you think about why is the bioprocess so expensive, because the reason is that R&D cost is very high, process development is very high, manufacture process is very costly, and quite often you fail. If you think you have 10, you develop 10 products, 9 failed, 1 success. So the price setting for the 1 products, you have to cover the cost of other 9 failures. That's why it's making it very expensive. So how we actually get it cheaper? So we should, because this is the reason, biological data is very scattered. This is a lack of quantitative prediction. We know if we learn physics, chemistry, even chemistry, and we write equations. You know you can predict. If you do heat transfer experiment, you say the error, it should be in less than 0.1 degree centigrade in prediction. But here is very scattered. We can't predict actually quality inaccurate. That's why a lot of them depends on experience and expert. Okay, expert. So-called expert. If you think who is the best expert in the world, okay, we got AI now. We can get would be super expert. So I give the example why this complication. So growing cells, I don't know, probably in the audience, some of you grow cells. It's magic. So why is magic? She said, actually, you look at the protocols. This is adding this, shaking, centrifuge, whatever steps. You can do it, no problem. I cannot do it. Cells, I follow the example. You follow the protocol, the cells not grow. I quite often ask my postdoc, I said, what I did wrong? They said, you need to play good music to the cells. But that's a joke, but it's really as art, as a skill. So you look at the data, how scattered the biological data, right? Look at the right one. This is actually the insulin response to the drug, okay? The drug for diabetes drug testing. Insulin response to glucose concentration. You just look at the standard deviation. You see, in any of the physical engineering experiment, if you present your data like this, your supervisor will kick you out. But this is biological data. It's very scattered. That's why if you design the process based on that data, so which point you rely on. This is a complex imaging because the people in biology and cell biology to see to believe, right? You see it. Then I want to mathematically describe the cell morphology. I'm sorry, this video not play, but then I want to describe that. How you describe is not a simple sphere, not a square, not a whatever shape. You see how you describe that. So I know a lot of people here, a lot of them are interested in neuro. So some years ago, we did this iBrain project. We like to bottom up building human artificial neural network, human neural network. So start from human neural stem cells. And then we have multi-layer structure. You have input, the middle layer, output. It's quite complicated. Take 5 years to make this. But what I want to show you is actually when you get this is the left side is illustration. You have input, signal going down, you have output. Then we use this microelectrode array. So take the data reading out. So later you want to theory, we want to retrieve it. What is the neural model here, right? Whether the artificial neural network, this model, this E function makes sense or not. That's what we wanted to do, but we got other data. We think, how complicated is this data? Who can analyze it? So we still have tons of data there. We still haven't analyzed it. So because this is beyond our individual human brain. That's why we need this super expert, super intelligent. So that's AI, right? So that's, and then you see, if we got this, all the cellular products, tissue engineered products, cell products, or any biological, the genetic products, and then we got the products, how we store them, how we transport them. This is our engineering issue, right? So then you see people will say, you just simply freeze the cells, put in the liquid nitrogen. But it's not that very simple. So people always say individual cells, there's a survival window for freezing rate. You have to freeze that at a particular rate. And you need a cryoprotectant to protect them. And then you have the challenge for if you got tissue, is a three-dimensional structure. And you have heat transfer, mass transfer, you have coupled, if you got the freezing, and inside the cells, outside the cells, multi-scale process. Yeah, very complicated. So the bottom line at the moment is still try and error. They said, okay, and because people will say, I'm just adding DMSO, but then clinically you cannot use a lot of DMSO because you need it to be, that's will cause response in the body. So it's all like, at the moment, is a black box. You need the luck, basically. So this is a human embryonic stem cells. Many years ago, my student did that. This is commonly now, this mesenchymal stem cells. You think it's sometimes the work, sometimes doesn't, right? So you see, there's no guidance, no general guidance. This is again, I think if we have a superhuman can help you say, do this, adding this will work. Okay, so that's actually then we do not need so many PhD students to test. So if you want to completely get rid of the cold chain, you can use freeze drying to dry the products. So this is like the process, and then you have during the freeze drying, this phase change. This is, I think we have Professor Larry Chow there about the chemical technology, right? This is you put the drug, you have either the protein drug or virus or vaccine, and then you formulate them. When you freeze them, drying them, and this phase change, they all push it around. And this gets to molecular level. So what chemicals to add to protect the structure of the biologics? Because the structure linked to the function. You think about protein, if they denatured, and then they do not have function anymore. So this is actually, if you look at what happened, this is in the bright field, you see this is during freezing, freeze drying, and this is what happened. And then if you do this colored multiphoton imaging, you can see this during the freezing and the freeze drying process, all the molecules rearrange themselves. If we actually can identify what molecules to protect our biologics, okay, to make sure they do the job. And then, so if you below that, if you magnify that a bit, you see this is very nice pictures. But how you model that? You see, we engineers, we like to model that, we predict it, and then how you do it. So this is, again, this freeze drying is actually a act at the moment. So this is a bit. So we want to get, if you can get away with the cold chain, because the biological stuff, you have to freeze them, it will make a bigger impact. All right, so this is actually the one. So there's a magic molecules related to this freeze drying, is actually trihalols. Okay, trihalols is a sugar, a kind of sugar. This bug, this is a little bug, right? It's actually, they are in the Africa desert. They can get the bone dry, completely dry. And but when you get water, anytime they touch water, they come back, they crawl away. And the first discovered in New York Museum. There are these people collected this in Africa, stay in New York Museum for 150 years, and in a glass capsule. And one day the technician accidentally break the capsule. This bug jumped out, not jumped, basically when you break, they just lift up, and happened, landed on a drop of water on the table. And amazingly later, this bug starts to crawl. This is later generated a lot of interest. They said, okay, what it is, and find out inside the cells, there's a lot of tree halos in it. But the tree halos cannot cross our cell membrane. So then this, we're trying to say how we get the tree halos into the cells, then to enable us to freeze dry them. So that's why we have we have few patents on this, try to get tree halos into the cells. So I come back to this. This is to come back to say why we need AI, right? You see, basically when we beyond our appreciation, so many data, there's not precisely predictable, a lot of uncertainties, but a lot of data around. And everybody telling different story. So we need a superhuman to sort this out. So that's why if we can use the super experts, that is AI, we can maximize the likelihood of success and quickly. Remember, if you get early time, get to the market, you actually will make quite a lot of more money because the patent only have limited time. You know, actually, if you think of the pharmaceuticals, which drug make most of money, the biological part, is EPO. This EPO is a protein drug and make most of money. But then when Amgen expect they will get approval from FDA, they said that they approved, we should as soon as possible launch a lot of products in the market. But the manufacturer need a fermentation. So you need actually, you culture the cells, you need the weeks. They said, how we do that? How we speed up? The time actually for the manufacturer is the fermentation, grow cells. The complication, costing part is downstream separation purification. So what they do, they culture cells, and when you get this products mixture, they don't do any processing. They freeze them, get big ice blocks, leave in the storage. So when they know, okay, FDA approved, so simply purification launch the product. So it's actually because you only have 20 years protection and patent. The development, you probably used 18 years already. So you only have very limited time. You have monopoly to charge a lot of money. So speed is very important. So what is AI? I said we need AI. I'm not an AI expert. Okay, I'm not an expert. So I never, well, I think I said, what is AI? So I said, I asked DeepSeek, ask ChatGPT. Well, basically it's understand language, recognize imaging, and learn from experience, make a decision, solve problem. I think solve problem. We have the problem. You see, that seems very interesting. And so this is what is AI. I think, what is not AI, right? It's very interesting. If you look at what is the response, so DeepSeek said, that's a sharp question, right? So basically, if you're looking at what is AI, what is not AI, so simple automation is not AI. Basic algorithm, if you program it, they do exactly what you ask it to do. You defined it. That is not AI. Rule-based system is not AI. Fixed response, not AI. Traditional search, so this is not AI. Then I said, I'm not a computer scientist. So what related to us? I said, okay, what about, so this is again, ChatGPT basically gave you a similar idea. What is it? Not AI. Okay, so come back, programming, statistics. Okay, it's not AI. So human controlled decision is not AI. So this is actually quite a, you have to understand what is AI, what is not AI. But then I said, I'm not a computer scientist. This does not make us very clear cut to me. Think about my profession. I want to design a process. If you got the process design, process development, and the control, is that AI? Process design control, right? It said process design is not AI. Control is not, rule-based control is not AI. Fine, that's not AI. But then if you got the process design with AI, it's become, yes, it's AI. And the adaptive control with AI, machine learning, it's AI. Everything could be AI, right? You see, what is AI? So I think as a founding body, I was told, applications for research funding, 70% of the research new applications got AI in it. They all claim it's AI. So this is a wave. Everybody can say, I'm doing AI. I said, so if you think with AI is AI, I suddenly realized I have been doing AI all these years, you see. I have this is actually 15 years ago. This is a few publications then. Basically, we're looking at the stem cell differentiation. Every lab has a different story. So I have this student, Wei Wang. I said, why don't looking at this thousands of publications, looking at what we can learn from it? Why we have to do experiment? Can we actually do digital experiment? All right. We got these conditions, stem cells will turn into bone, turn into cartilage, turn into muscle. He said, oh, fine. And he went away. During his PhD, he published 8 papers in one PhD and graduated from Oxford. So that's a few publications. This is for machine learning thing. At that time, of course, we do not know this is AI. We just say it's a useful tool. But this student, this which I must say a few words, during his PhD, he published 8 papers, right? He also wrote his first book. So that's his trilogy, and they get the first book become Amazon's bestseller. Is a sci-fi book. Then he left Oxford, went to Brown University postdoc, and then got to Stanford. And he gave up research. Then after that, he became this so-called data scientist and a sci-fi bestseller book writer. So he wrote 3 books so far, and a lot of people chasing him to make games based on his book or making films. I thought he's you see, completely different approach. But that is, you see, I said, oh, we actually did AI at that time, you see. So now people say AI can predict our stem cell differentiation. We already done that 15 years ago. We proved it work. But of course, now you got a lot of new data. You got a better prediction. That's a AI can be, there's some low-hanging fruit. It's very easy to understand. For example, pathology. And then you got a picture, whether you need to see whether you got a slice, you said it's got cancer or not, and what type of cancer, right? And what grade of the cancer. So that's routinely done by pathologists in hospital. But for animals, for wet, there's not many pathologists. Okay, I was told in the overall world, no more than 400 people qualified, certified as an animal pathologist. And it's actually understandable because you see the pets include the cats, dog, turtles, snake, birds, they're all different. So that's why, yeah. Okay, that's why so you could now use this by AI. You can get AI-driven innovation by 3D printing blood vessels. You see, you can design on this because you now have so much data. You can say, what would be the best? Let AI to design it we don't need it's a black box. Here's the data. As long as your data are good data, because garbage in, garbage out, right? And so this is my colleague Wei Huang designed this SimCell platform. You got the cells, you can get all the functions, and finally you get rid of genomes. say they could not replicate, then it's safe, right? Because they couldn't replicate. You just use the shell, and it's actually quite a useful platform to do various things, to do vaccine, to do cancer therapy, and bacterial therapy. That is all that engineering biology. So this is actually, I'll ask the AI person. We have a lot of people in Oxford working on AI. I said, I thought that what AI can do, because I thought the AI, computer people, you have all these models, but then you need to train the model, right? So you need the data. So if garbage in, garbage out. So this is why we need the good data. And a lot of them, data may not exist. So this is why we need engineers or technologists to develop sensors. And so what a sensor, what parameters you need, how you measure that, and how you feed into your AI model. So outside the sensor part. And more importantly, we also need the activator. So basically, we need action. Okay, auto drive cars, it's very easy to say, you see, the car is auto drive. But for our discipline, for what we want to do, you need action, how you act. So that's why you need to the activators. So this is actually, give two examples. One is actually 3D bioprinting, right? So this is, I got the slides from Professor He Yong in Zhejiang University. So 3D printing, you can print cells, you can scaffold, everything is one activator. If you feed the model, they print as it is. And I also have Haitao sitting there in your university, and he work on this robotics for this actually experiment. So you need, because we said human error is one of the main issue in this process, because so many steps. So if we got all the robotics, AI help make a decision, that would be the way to do. The bioprinter, you can make it very, very complicated. This Professor Hua Yongyang also in Zhejiang, and they got the prize. They can do 600 nanometer resolution. So you can, but the cells is 10 to 20 microns. So this is subcellular level, you can print it. And again, this is Haitao's data. You can combine this, you can replace postgraduate student, the try and error. Basically, we try to replace humans in the process. You go to automation. You need AI is brain make a decision, integrated with robotics, and then you get everything done that way. Much quicker, reliable. I think so in future, I would say personalized medicine, just like in the morning, you wake up, your room, your bathroom, your sitting room, your bedroom, full of sensors. Say, how are you today? Oh, you need more of this vitamin, you need more of this formula. You go to your AI system, will all got it up. That's cook your breakfast soup there, or magic drink, you take that. That's personalized medicine and the healthcare system. So AI can manage our health one day. That's a coffee machine. I'm not doing advertising for them, just an image from the web. I think that's the last one. Thank you very much.
Ladies and gentlemen, my name is Larry Chow, head of the Department of Applied Biology and Chemical Technology. And first of all, thank you very much, Professor Cui, for giving us a very enlightening lecture, particularly putting two very hot topics together, the AI and the biomanufacturing. And also I'm really impressed with the really wide range of research topics that you have covered in the lecture you just have heard. So in the next 30 minutes or so, what we're going to do is to have an open floor question and answer session. And so the floor is open. If you have any questions, please raise your hand. And then we have some workers, helpers to give you the mic. Okay, thank you.
Well, first, thank you very much, Professor Cui, for really exciting talk. You know, I am a layman in this field, but through your lecture, I learned quite a lot of this outstanding work you have done. You mentioned about this AI, but we know that when you want to use AI, you need a very good data, right? You also mentioned that if the data is not good, then will be garbage in, garbage out. But by looking at the data, I mean, you have only like a tissue growth and so on. Those, you also show there's a very big scattering of the data quality. How could you train the AI, I mean, good AI, by using not so good data? That's my first question. The second question is about the tissue engineering. I mean, it definitely looks very exciting. But when you try to modify like DNA, things like that, so how do you ensure the safety of the human, the patient? I mean, definitely we know that some of the DNA modification definitely produce a very nice result, but a lot of not being tested, right? I mean, that's why like US and the government are very cautious in approving those technology because you need a lot of clinical trial data, and which might not be available. Thank you very much.
Okay, thank you. I think, yes, for garbage in, garbage out, this is state. For biological, I think we are very lucky to working in this cell, you see this tissue engineering stem cell area. We're working on downstream. We're lucky in a way. This is a very, very hot topic in the last 20 years, 30 years. There are a lot of cell biologists working on this fundamental science aspect of research, study cells. So indeed, actually, one of the key issue in our work about 15 years ago is how to deal with the scattered data, how to, the missing data, quite often missing data, very scattered, the missing part of data. You hardly get one complete set of data. So then this is actually all the computer people with computer algorithms try to identify, say this is this is off. You can identify their bad data, basically, eliminate that into the system. So a lot of them is to establish the database. That is how to evaluate that. Indeed, that is a very critical question, but I don't, so basically there is a basic, if you see at that time, we said, okay, we have this one, this does not look good. Then you have the human intervention to see actually, probably it doesn't, it's not. Then we just put a mark, do not include that into the database. So I think that's a key, but this is why, but this is again, you have human intervention, but the AI may maybe find some way to eliminate that or modify this, adapt this. Because this, I hope AI can deal better with the missing data and the incomplete data sets. So that's the first one. Second one, this regulatory approval is very critical in this area. You know, in China, there was a few years, 2015 before, and very active in stem cells research, and people trying to sell you see, and to get the infusion of your stem cells. And then until we just see that person died during the process, and then China government completely banned it. So until now, started to relaxing, but it's still with tight control. I think control is very important because I visited many these cell therapy centers. Basically, it's all done by clinicians, medical surgeons. And they said that they have few students grow cells and he put it into the human body, right? It's just like when I visited them, they said, look, this is our incubator. So you mark the patient 1, patient 2, patient 3. If you mix the cells, the patient will have a bigger problem. My students, they are very good. They never mix it. But that's not the way to commercialize to a large scale. For real manufacturers, this is a standard operation protocol is critical. So I always gave an example where I said, look, if you go to Chinese restaurant in the world, any Chinese restaurant, they all serve fried rice, right? Egg fried, Yangzhou chao fan, everywhere. But they all different. Every restaurant is different, right? Depends on the chef. You go to McDonald's, you ask a burger, anywhere you got the same burger. So this is why I think if you want real industrial scaled production, you need to go to McDonald's model, not fried rice. Okay.
Thank you. Any other questions? First, actually, I have a follow-up question about the, you mentioned about the regulatory importance. And you mentioned about some issues in China. Can you give us some comparison, let's say the regulatory control in UK versus, say, in China? Now, I work in Hong Kong, and I also, at PolyU, we actually also have a ATP production facility. And in the past few years, actually, we have gone through very, very difficult job in getting the regulatory approval. And I would like you to comment on the sort of balance between, on one hand, there are a lot of patients waiting for us to, save their lives. But on the other hand, we also have the regulatory people who say, okay, slow down. We need to be careful and patient. And then we also have the scientists in the universities and all of the postdoc research students in here, and they just keep pushing out more new discoveries. So how are you going to balance all different parties? Do you have any insight on which system may be better, advantages in UK versus, let's say, in mainland China?
Well, that's a bigger question. I think actually, as a scientist, nobody likes regulatory control, right? Because this is great. But then from the customer, from the patient point of view, is actually very critical. You see, if you got the medical people, the first thing the doctor said is safety, patient safety is a top priority. Right? So I think that my piece of advice is that I try to work with medical clinician, because they know actually how to put this into patient. And then that is, I think in this actually apply to patients, there's two distinct difference. So one is, if you look at the medical people, what they want the first, medical research, they want the first on man. They said, I'm the first did this. So then, but then they do not, quite often, they do not need regulatory approval because they treat that as a clinical case study. Okay, so they got ethical approval in the hospital system, they can do it. But then that's not, they can't charge, you can't make money. It's a research called the clinical case study. And that is relatively easy if you can working with a good surgeon, a good medical clinician. But that's important to us. But then when you transfer it to industry, you want to make a lot of products, make commercial success, regulatory is really, really important. Otherwise, you can't do it. You can't scale it up. So related to the regulatory bodies is very difficult to say because they're all changing, right? FDA actually is quite sympathetic and willing to, working with scientists to absorb the new technologies. And they have, for example, they set the regulatory for mesenchymal stem cells, for example, quite a few years back. EMA used to be in a mess. It's a European one. It's quite nobody knows how they work because they keep different languages. People argue about it. UK is in a particular mess because they left the EU. Try to set up their own regulatory body, but then nobody knows how this works. Basically, at the moment, still copy what we have before. And but the UK authority, UK government, they very encourage new technology. So a lot of this new advanced one, a lot of controversial one, and the UK approved it, and not in the US, not in the EU. I'll give you an example. Now UK approved this mitochondrial treatment for embryo. If you have a woman and they have a genetic disorder, they are mitochondrial, so if they got a new baby, the baby will have the disease. So then you can have another woman donate an egg and get the mitochondrial, replace mitochondrial of the first woman's egg, and artificially fertilize, use the father, and then get a new baby produced. So that's we never approved in the US, but it's approved in the UK. So that is, so UK is sometimes lead in some areas. But the actually in the EMA and the EU, the regulatory framework is almost the same. If you got one approval, very easy to get the other one because all the document. And the difficulty is whether they accept your data, right? But the EU and the US, I think they have easier to accept the data from the other side. But China is quite different in a way that a lot of data, clinical trial in China, they may not accept. They may say you have, okay, you can come to the US, but you have to do the clinical trial again. That will push a lot, a few years. Each clinical trial take years, a few years. And probably until now, I think China regulatory body probably the most conservative one. If you got a new, this is actually, I think it's changing, but certainly a few years back, if you submit, anybody submit a new, ask for a new approval, the first thing they ask you, has this approved by in US, FDA? Has this approved in EMA? If you said yes, FDA approved similar things, then they can start looking for that. If you haven't, they said, we probably do not want to be the first one. So that's is, they always ask that. So it is, because actually from engineering aspect, the large scale manufacture regulatory is quite important. I give you, and also the interesting is to involve, I always say the scientists, the clinicians, engineer, we should be working together. That's the first thing. Second thing is to get earlier, get involved with regulatory body. To then you can easily, to later time, you do not have to redo a lot of stuff. An example about this, I mentioned this EPO, they make a lot of money, but when they rushed into apply regulatory approval, and they're based on laboratory process to rolling bottles, cell culture rolling bottles. They use this process applied for regulatory approval. And the FDA said, that, yes, you get approval. Then you have to manufacture following what you apply in the application. Then they cannot use bioreactors. They have to use rolling bottles. So how you make a lot, use rolling bottles. So they got a contractor in the UK called the Automation Partnership. They said, can you automate the whole cell culture use rolling bottles? So they have this big room full of these bottles rotating, 1 million bottles rotate. So you have robots, one bottle, change the medium, put it back, one bottle. So that's, they managed to get away, but they have to stick to what they apply. You see? Okay, thank you.
Thank you. Any other questions? Okay.
Oh, thank you. Because I'm just a freshman from ABCT, so Professor, I don't have the sufficient courage to ask you some frontiers. But as a freshman, I realized a question that because AI and the other technologies brings us a huge change or revolution. So I think our education needs some revolution, but maybe it's not get the step of the other the technology revolution. So I want to ask you that as undergraduate, what I should to study except for the traditional study methods. Thank you.
Well, that's I think the other day I was talking to the president of universities. I said, this is actually the I think education, how we teach in universities. That is actually a bigger question, right? Think about the COVID, the 3-year COVID. Actually, a lot of no lectures. We have recorded the lecture in Oxford, and you never see the student, and they got degrees, and they probably doing equally well, right? So this is now with AI, a lot of this knowledge explosion, and you get iPad, you got everything. right? You can get lectures from MIT, you can lectures in Oxford for same topic. And they probably better than some other lectures. So it is actually a bigger question. But then let's come to what's university for? So why you're here? You see, it's not just the learning process, it's not just attending lectures. It's a way you have to think, right? So we keep telling our students what's a basic quality for important part. One is critical thinking. So it's just critical thinking is not just people telling you, your lecturer, your professor, your AI tell you this. And you have to figure out whether that's true or not. Because most AI stuff, you go to DeepSeek, you ask the GPT, and the answer is not quite often not accurate. Okay, you can't say sometimes it's totally wrong, but it looks like very logical, but it's a completely garbage. And when you go to the scientific part, what I find is very good is polish your English text. It's very good. Right. So that's why I think that's a critical thinking. Second thing is the independence. So you need to grow, basically get mature. The education is university life is a process to grow up. Right? So that's get mature and become more and more independent. Related to what you learn, actually, you see, you have to learn something because your subject. Actually, what you learn the subject, not necessarily that critically important in your future career. As long as you learn properly. I think you see, this is why the transferable skill is important. If you're trained as a chemist, what I find is trained as a scientist and trained engineers, actually the way of thinking is different. But then it doesn't matter. It's trained with civil engineer and the chemical engineer is different as well. But then every discipline, they are fully established over decades. They have this methodology, the way to analyze complicated issues and the processes. So we learn that, and later you can apply to different fields. So this transferable skill and the systematic training of a particular discipline, it doesn't matter what the discipline, they all established. Then that, I think that this is very important. But this is my personal view. And but you have to really equip you to face the real world in the future. But then you learn mechanical engineer, not necessarily you will go to manufacture, right? You learn civil engineering, you may not do the building environment. So but you learn this how to approach problem. And I think that is important, this transferable skills. So later you can do whatever. You see, I think our student in engineering, 40% of graduate on day one, they go to financial institutions, business consultant. So at the beginning, I said, I spend so much time to teach you engineering. You go to finance, right? And then later I realized if my pension fund managed by engineers, far secure than bankers, economists. Okay, thank you.
May I also add that actually we have a first-year undergraduate students who have the courage to stand up and raise questions about an area that he's not familiar with. And may I say that this is one very important character that it will be important, will be for the future success for every one of us to learn. It's not just the AI, but the courage to ask questions. And now AI have everything, but you need to know and have the courage to ask. And that's actually very, very impressive. Okay, thank you.
Okay, I'm CUI Zhu. Actually, I've only been to PolyU for 2 weeks. So it's very interesting to attend this peer seminar and very enjoyable. Although I'm completely in different field because I come from EEE department. And I would like Professor Cui to give comments on 2 scenarios which I'm doing, and also I may have a suggestion, could be completely wrong. One is in my field, one is to do the design optimization, for example. So we're already using the AI type things to do the design optimization. But very often towards the end, we still needed to select by our eyes and using human interference kind of thing there. But from what you said, this probably is not belong to AI, or at least not completely AI. That's one. Second one, I've been involved with manufacturing tolerance inference. There's huge numbers and to deal with, and particularly try to identify which manufacturing tolerance could be dominating to the quality of the manufacturing, for example. And then my student has been using AI at the preliminary stage, but to the final stage, we have used our own model, which is more suitable and for the very limited number, for example. So I would like you to comment on this too, but then I'm only, because I know nothing about AI, by the way, anyway, and then I'm only also guessing, is the AI necessary to go through all from right beginning to the end? Can we divide that to say like a hybrid way, AI and a more conventional model or human interference? Thank you.
Well, thank you. I think that's a very good question. Let me ask the last one first.
Yeah.
I believe AI is, I think what we do eventually all hybrid model. I don't think you will have AI from beginning to end. I think if we can do it, properly, why we need AI? Right? If you just said 1 + 1 = 2.
But if we can do it, we can have a cup of tea. It's better.
but I think exactly we should leave the. I think probably most of them is a hybrid model. You see, if you can mathematically describe it, precisely predictable, why you need AI? Put it that way. And so I think it would be hybrid models, and you live in the, because what I think of my part is actually when you got a lot of scattered data, uncertainties, you can put a lot of data in it that they can make a better decisions than us, than the human. That's the last one. The first one, I think AI is, AI is after all is a tool. I think AI is a tool. People say AI will replace human. I don't believe it. I think if to get that stage, our existence is under threat, right? So why we need a human then? But AI is a tool, so it's just like where we use it, and then use it wisely. I think that's important. That's a bit. I think for your second one, I really don't. It's too technical. I think you see, it's just this. I can't give you any suggestions. But then, what I find, if you have a lot of data, a lot of uncertainty, you just say every time you're looking at this, your brain will explode. Why I started this? Maybe you can dump into a black box to see something useful come out. You see, but you see, I think AI is just a tool. But at the moment, is everybody waving the flag of AI? But then we really needed to get AI do something useful to us. If we can do it ourselves properly, why you bother with AI? So you can predict.
Thank you.
Okay.
Any other questions?
So thank you very much for your presentation, and I'm particularly curious about the AI part. And in biological research, we often encounter various type of data, such as images or number of values. How should we handle this heterogeneity in data when we training the AI model? And regarding the performance of AI model, how should we evaluate it? And how can we explain to biologists why an AI model makes a certain decision? How can we ensure the interpretability of the AI model? Thank you.
That's a Okay, that's a very good question because I threw out the problem. I did not give answers because I do not have an answer. I think that is actually a proper, at least one or two PhD thesis to work on that. Because this imaging is a biological imaging, it is quite complicated. And then, so if you know exactly what you want. For example, you got the pathology. You said, I want to identify cancer cells in this slice. Very clear, defined objective. Is okay. That is doable. And then if you got just, I have all the question I threw out is that I have these cells in all the shapes. How you describe it? How you link to their cell fate, a different stage? Then I do not have answer yet. Okay, but it's a very good question because the imaging, yeah. The interesting part about the AI, the machine learning and the imaging analysis, imaging is because that's in GPT even said, this is AI. So then it should be core part of imaging analysis and machine learning. But how we, I think we need the people looking at the individual. So this is a bit about AI. AI is a tool. It can apply to any different scenario. So if we actually, you either reject the tool, I said, I don't want this tool, or I use this tool. So I think if you can, it's always, it doesn't do any harm, try to use it to see whether it can help you, right? So when you explain to cell biologists, the bio people, actually you don't have to explain to them because they are very keen on AI as well, many of them, because they face the same problem. We said, this is a problem. We can't solve it this way. Can we try this? So that maybe you can do it together. Okay, thank you.
Okay.
Sorry. For you, BME student, I study, sorry, I also study about CAR-T in a class. I have some questions to ask. First is about how you bring the research to real manufacturing. Because, for example, there's a lot of research in PolyU. I talked with each professor in BME department. And I found the main problem is we do a lot of research, but execute is a problem. When we try to do, for example, like you collect the data, but when you try to make it effectively, like we have AI, but the problem is AI is generally about the methodology that you, how to analyze such a data. Even the studies, they're partly for example, like computer-based, or because I'm IT-based, but how you apply using the AI technology to set each rule, and even how you develop some new rules by the AI to make the research effectively. Second, for example, like about the manufacturer. For example, like yesterday I go to Beijing University, I see some about the lighting in Dongguan. There's some faculty in Dongguan. They have some about lighting problem. They do it very deeply for each material and make the manufacturer more effectively. But actually for nowadays for manufacturer, we are stopped to do how to make the manufacturer effectively. We always rely on the machine aboard from like UK and US. But how in our nowadays we know from many professors, many different area experts, but how we cooperate together to build a better model for manufacturer. Otherwise, our cost will not be going down. And about the research of the patient, can we suggest your government, for example, like some patient may be more serious and they may be no hope, something like that. We need to, how we change the law or such a government to collect more of the data, to maybe try to apply to them and give them at least our hope, rather than just research based on the research level and doing our research, publishing, and then we have nothing to do with the society. So I'm asking Professor, how do you execute about this? Thank you.
It's all bigger questions. I think the second question is that actually this is exactly, I think, why you have a pair. You have the PolyU Academy of Interdisciplinary Research. You can get professors from different discipline can working together. You see, that's what you need, this type of platform. But then what you are saying is on the government level, but that's would be beyond that's actually, I think the Hong Kong get this, UK or Hong Kong, Hong Kong is all these big programs, and many of them I see is interdisciplinary. So that's why they encourage people from different disciplines working together. So in your university as a pair is a very good model for that.
Sometimes they didn't know each other area. For example, like you tell some AI expert about the biomedical is very difficult. So I think that sometimes we need to find some platform or some cooperation that we need to a little bit more thorough about each area, and then we cooperate together rather than just put the data or something like that. I think that maybe is the main problem is about the research of currently biologic area. I see a lot of professors, they tell a lot of strategy, but I never see they execute. I'm sorry. Yeah, because I'm the one why I study PolyU, I feel business, but I want to really execute about the project, not just doing a publishing and get a high ranking of university.
Yeah, I think there's well, one thing is that you could stand up and say, I organize this lunch or a brainstorm session, and people can join in. You take a lead to have a special topic. Let's discuss this one. That's a one way to get people get together. But I think answer your first question. Also, you also mentioned is that what job we should do? What is our job? What is other people's job? Because actually, we are in university. So university, you do teaching, of course, that's education, training, and do mainly thing is we work on research. So you got research, and the next stage, development, and the next stage, innovation, and the next stage, commercialization. Then you serve the society, provide the products for the patient. So we at the university, we are not in that part yet. So this is why important thing is what we do is push the frontier. You see, we want to do something new, something. We are in university, we got a salary, we can afford to dream. Okay, why we don't do this? Industry people, they have to sell products to get a salary, so they working on the close to market. So this is why we said that, yes, we do this. We write a proposal. We said that this will potentially serve 100,000 people, but that's actually is not our job. Our job is to develop the technology, prove the concept. Then you go to high-tech companies, you set up companies, scale up, and then to the manufacturer, then that's the company grow, that's a company activity. So I think we all encourage our people think about making money, commercialization, but actually we should separate what is R&D and what is commercialization. And then we probably better do it in the early part, not the later.
Okay. Thank you, Professor Cui. I think because of time, I can see there are still some students or staff who would like to ask questions. May I suggest afterwards we can have more discussion with Professor Cui. But may I now invite Professor Chen to present a little souvenir for Professor Cui. Okay, thank you.
All right. We also like to thank Professor Chow for moderating this session. And I'd like to thank everyone who come here today, attend this seminar. You probably know that the PAIR organize distinguished lectures, seminars from time to time. Normally, we only invite each person give just one distinguished lecture for when they come first time. when they come the second time, then becomes a seminar. That's why Professor Cui today gave this seminar. So I believe you probably will receive a lot of announcement from peer, and I encourage you to attend our future lecture and seminar. So thank you all again, and this seminar is adjourned.